Hello CSIR NET aspirants, viewers and subscribers of Biotechnica. I'm Dr. Tanushree and I welcome you all to the video on CSIR NET Life Science Unit 13, that is Methods in Biology, Important Previous Year Question with Solution Series. In this video, I will be discussing previous year questions on topics restriction enzyme, DNA manipulating and modifying enzymes, and restriction digestion. The topics from Unit 13, Subunit A, that is Molecular Biology and Recombinant DNA Methods. If you go through previous year question paper, you will find that the questions are most frequently asked from these topics. So while preparing for CSIR NET exam, make sure you emphasize on preparing these topics. Now let's have a look at the type of previous year questions based on these topics. The very first question is, given below are names and recognition sequences of a few restriction enzymes that are used for cloning experiments. The cleavage site of each enzyme is indicated by symbol *. So they have given you several restriction enzymes and their recognition sequences. Also given below are different vectors shown in column A and insert fragments shown in column B generated by digestion using above enzymes. You have to find out which one of the following options represents the correct combinations of the vector and insert respectively, which generate compatible ends for ligation. Now to answer such questions, you need to be familiar with what are restriction enzymes. The ends they generate when digestion is performed by using them. So you need to be familiar with blunt ends, you need to be familiar with sticky ends. Also, you need to be familiar with the concept of vector and insert joining in cloning experiments. So let's first understand what are restriction enzymes. Restriction enzymes are also popularly known as molecular scissors, and they belong to larger class of enzymes called nucleases. Nucleases are the enzymes which break the phosphodiester bond of nucleic acids, and they can be exonucleases and endonucleases. So basically restriction enzymes are endonucleases as they cut within the DNA molecule. And the interesting fact about restriction enzymes is that they don't cut randomly. They recognize a specific base pair sequence in DNA and cleaves DNA within this sequence. So here you can see an example of restriction enzyme EcoR1, and this is the recognition sequence for EcoR1, and this is the site at which they cut. Now different types of restriction enzymes are available, type 1, type 2, type 3, type 4. But mainly in recombinant DNA technology, we use type 2 restriction endonucleases. So basically restriction enzymes, as they are endonucleases, so we can also call them as restriction endonucleases. And here you can see the examples of restriction enzymes, the organisms from which they are isolated, the recognition sequences of these enzymes. So you can see the recognition sequences. For some it is 4 base pair, for some it is 6, for some it is 8. So the recognition sequence for restriction enzyme vary from 4 to 8 base pair. And here you can see the ends they generate on digestion. So some generate sticky ends, some generate blunt ends. Now what are blunt and sticky ends? Let's take an example of enzyme ALU1 and EcoR1. ALU1 recognizes 4 base pair sequence and EcoR1 recognizes 6 base pair sequence. Kindly note these are palindromic sequences. So type 2 restriction endonucleases, they recognizes palindromic sequences, which are the sequences read same 5' to 3' direction of both the strands of DNA. For example, EcoR1 sequences, recognition sequences GAATTC. So 5' to 3' direction on one strand, it is read as GAATTC, and on another strand, if we read it again from 5' to 3' direction, it is read same, GAATTC. So these are palindromic sequences. Now coming to the ends they generate, so ALU1, it cut both the strands at the same place. So it cuts at the same place in middle of the recognition site and produces blunt ends. So blunt ends, they don't have any overhang region. On the other hand, EcoR1, it does not cut both the strands at same position. It cuts on one strand, let's say between DNA. So on another strand, it will cut several nucleotides away from it, resulting into overhang regions. And the ends are called as sticky ends or cohesive ends, as they can stick back by transient base pairing, like A can pair with T, T can pair with A, and so on. Now, it is very important to note that while using the enzymes in cloning, you have to use the same enzyme to cut the vector and insert to generate compatible ends. Kindly note, the enzyme will generate blunt ends, so any blunt end could be compatible with another blunt end as they don't have any overhang region, so they have to be simply joined by DNA ligase enzyme. On the other hand, if we are using an enzyme which generates sticky ends, so usually sticky ends are much stronger than blunt end because they can join back by transient base pairing. So in cloning, we have to use the same enzyme to cut the vector and insert, so that compatible ends are generated. Also, you can use the combination of enzymes which can generate same type of sticky ends like BAMH1 and BGL2. So GATC, let's say if we digest the vector with BAMH1 and insert with BGL2, then you can see they generate compatible ends which can be joined back by transient base pairing. Now, as we are familiar with the basics, let's solve the question now. Always remember, you have to use the same enzyme or combination of enzyme to cut the insert and vector, which can be plasmid, cosmid, artificial chromosome, or bacteriophage genome. So here we have used EcoR1 to digest the gene also, the insert, and plasmid, that is the vector. So to use vectors and insert fragments for cloning, they should have compatible ends. So as shown over here, the target gene and the plasmid, as they are cut with the same enzyme, they have the compatible ends and they can join back by base pairing. So our target gene is inserted into a vector and the recombinant DNA molecule of plasmid is formed or generated. Now, as we are familiar with basics, let's solve the question. Given in the question, the combination of enzymes used to cut the vector and insert, and we have to choose the combination of enzymes which can generate the compatible ends for ligation. So strategy for solving such question is that we will omit 2 options out of 4. So we'll start with option 1. So in option 1, A2 combination is given, and also in option 3, A5th combination is given. So let's check whether these combinations are compatible or not. Let's start with EcoR1 and HINC2. So EcoR1 generates sticky ends, whereas HINC2 generates blunt ends. So this combination is not possible because we cannot use the combination of enzymes, the one which generates sticky end and another which generate blunt end. So that means answer cannot be option 1. Now if we look at the combination A and 5th, that is EcoR1 and BG3, so both generate sticky ends, and kindly note sequences given are 5' to 3' direction. Now when both the enzymes generate sticky ends, we have to look for complementarity. So if the vector is cut with EcoR1 and the insert is cut with BG3, if they are compatible, their ends should be compatible. Now the BG3 3' to 5' sequence will bind to 5' to 3' sequence of EcoR1. So I will write down the complementary sequence. So that is T, C, T, A, G, A, and I'll just place it over here to check whether complementarity is there or not. So we can see that they are not compatible. The sequences are not complementary. So that means this combination of enzymes will also not be used. Therefore, answer will be between option 2 and 4. Now in option 2, let's check B and 2 combination, that is EcoR5 and HINC2. So yes, this is possible because both generates blunt ends and hence can be used for ligation. Now to further confirm whether this is an answer or not, so let's check D, BG3 and 4 BAMH1. So again here we have to check for complementarity. So I'll write it down the sequence of BAMH1 separately, that is G, G, A, T, C, C, and it's the BG3 3' to 5' sequence which will bind. So it is T, C, T, A, G, A. So you can see at least 4 base pair sequences are complementary. So enzyme cuts over here and enzyme cuts over here. So basically these are the sequences which are complementary. So yes, we can use this combination. And let's confirm about E and 1 also. So E is HINC2. E is HINC2 and 1 is EcoR5. So again, yes, they can be used as they both generate blunt ends. Hence, answer is option 2. Isn't it easy? If you know the strategy of solving the question, you can solve the question in very less time. So you need to be thorough with the basics, and also you need to learn the strategy of solving the questions in less time. Now another question is from CSIR NET December 2023. Question 2 is, which one of the following intermediate enzymatic reactions would be most effective in a facilitating ligation of blunt-ended insert fragment with a vector digested with EcoR1 restriction enzyme? So kindly note, the insert fragment is blunt-ended and vector is digested with the enzyme EcoR1, which generates sticky ends. So you have to find out that which of the enzymatic reactions given in options would be most effective in facilitating ligation of insert and vector. So option 1 is treatment of vector with mung bean nuclease followed by treatment with shrimp alkaline phosphatase. Second option is treatment of insert with clino-DNA polymerase and vector with polynucleotide kinase. Third option is treatment of vector with polynucleotide kinase and insert with mung bean nuclease. And option 4 of insert with clino-DNA polymerase followed by treatment with shrimp alkaline phosphatase. Now, to answer this question, you need to be familiar with DNA manipulating and modifying enzymes like mung bean nuclease, alkaline phosphatase, clino-DNA polymerase and polynucleotide kinase. So let's first understand the functions of these enzymes. Mung bean nuclease. Let's know about mung bean nuclease enzyme first. This is an enzyme which is an endonuclease. So this enzyme also contains a molecule, and as shown in the figure, it converts the sticky ends, the overhang region, into the blunt ends. So it catalyzes the removal of overhang, single-stranded extensions in double-stranded DNA. And it can remove both 3' and 5' single-stranded overhangs from double-stranded DNA to create blunt ends. Then alkaline phosphatase. Alkaline phosphatase is an enzyme which removes phosphate group from 5' end. As shown in this figure, alkaline phosphatase enzyme, it removes the phosphate group present at 5' end. So at 3' end you have hydroxyl group. So it removes the phosphate groups from 5' end. So this is useful when we have to prevent the recirculation of the vectors, linear vectors during cloning. And the name alkaline is given because this enzyme, it's active at alkaline pH. So that is why we call it as alkaline phosphatase. Polynucleotide kinase works just opposite to that of alkaline phosphatase. As shown here, at the 5' end, phosphate group is missing. So polynucleotide kinase, it adds phosphate group at the 5' terminus of polynucleotides or mononucleotides. So it catalyzes the transfer of gamma phosphate from ATP to 5' terminus of the DNA, bearing a hydroxyl group. Lastly, clinofragment. Clinofragment, it's a DNA polymerase. So basically, when you treat DNA polymerase, Escherichia coli DNA polymerase 1 with a mild protease, it results into two fragments, one larger fragment, another smaller fragment. So larger fragment having polymerase activity as well as exonuclease activity 3' to 5' direction. We call it as clinofragment. So clinofragment as shown over here, it adds the nucleotides. So it fill in the nick. So it synthesizes the double-stranded DNA from single-stranded templates. Also, it can create blunt ends by filling 5' overhangs, and even it can digest 3' overhangs to produce blunt ends. Now, as we are familiar with the function of enzymes, let's solve the question. Please remember the information given in the question, that insert is blunt-ended and the vector is digested with the enzyme EcoR1. So it has sticky ends. And as we have seen earlier, as we have studied earlier, that now as we are familiar with the role of enzymes, let's solve the question. Remember the information given in the question, that our insert is blunt-ended and the vector is digested with enzyme EcoR1 which generate sticky ends. As we have studied earlier, blunt end DNA cannot be ligated into sticky ended vector. So both insert and vector have to be either blunt ended or sticky ended. Let's look at the option 1. Treatment of vector with mung bean nuclease, which converts the overhang region, which digests the overhang region and converts the sticky ends into blunt ends, followed by treatment with alkaline phosphatase. So yes, this can be the answer because overhang in the vector, as a vector is digested with EcoR1. So definitely there will be. So overhangs in the vector after EcoR1 digestion can be removed by nuclease. And the phosphatase role is that it removes the 5' phosphate to prevent the religation of the vector end. So yes, this treatment, enzymatic treatment, can be done. Let's have a look at other options. So option 2 is treatment of insert with clinodNA polymerase and vector with polynucleotide kinase. Now clinodNA polymerase could also be used to convert sticky ends into blunt end by filling the gap. But clinodNA polymerase has to work on the vector because vector is having sticky ends, overhang region, not the insert. Insert is already blunt ended. So this is not possible. This combination is not possible. And even in option 4, it is mentioned treatment of insert with clinodNA polymerase. So again, this cannot be the answer. Then option 3, treatment of vector with polynucleotide kinase and insert with mung bean nuclease. So this is also not possible because our insert is blunt ended and we don't need to digest it with mung bean nuclease. Therefore, answer is option 1 here. Moving over to another question. Now this is one of my favorite questions from gene cloning and recombinant DNA technology. Most of the students, they find these type of questions very difficult because either they miss the details or they don't understand the question. They don't know the strategy of solving such questions. So don't worry, I will be telling you the strategy of solving the question and what points you have to keep in mind while solving such type of questions. In this question, a diagram is given which represents a 2 kb insert. So this is the 2 kb insert, successfully introduced in between 2 BAMH1 sites as shown in the figure. So 2 BAMH1 sites of a 3.8 kb vector in desired orientation. The Hind3 site on the insert and EcoR1 site on the vector is also indicated. So this is the Hind3 site on the insert and EcoR1 site on the vector. Now if the insert was introduced in opposite direction, this is a very important information. You have to choose which one of the following statements is incorrect. This is again very important. So strategy of solving such question is read the question carefully and try to understand it. If you don't understand it by reading it once, read it twice or thrice. Pay attention to the details, like what is already given and what you have to find out. So here the important thing that you have to notice is that already a figure is given and the question, the options which are given and the statement that you have to choose, it should be on the basis of when you are introducing the insert in opposite direction. So this means you have to redraw the diagram. You have to redraw this diagram which is already given. And another important point is that you don't have to choose the correct statement. You have to choose the incorrect statement. Now, most of the time students are in so hurry in solving the question that they miss these details. So please remember, read the question carefully, understand it, pay attention to the details given in the question and then start solving it. Now you have to practice of how to redraw the diagram. Most of the students, they don't know how to redraw the diagram. So I'll just show you. So this is the actual diagram which is given, actual figure which is given in the question. Now as mentioned in the question, this insert has to be in opposite orientation. That means this 1.2 should be here and 0.8 should be here. So we'll redraw it. We'll label everything as it is given in the question. So eco R1 site, and then 0.9 kb already mentioned in the question. This is the BAMH1 site. So insert is introduced between the BAMH1 sites. So let's say these are the two BAMH1 sites. And then this will be 1.2 kb because if the insert is oriented in opposite direction, and this one will be 0.8 kb. And this is the Hind 3. And this is 2.9 kb from here to here. Now to choose the incorrect statement, we have to go through each statement one by one. So let's have a look at statement 1. Digestion with eco R1 will linearize 5.8 kb plasmid. So it means that if I cut the circular plasmid from here, it will convert the circular plasmid into linear form. And this size of the plasmid, linear plasmid, should be 5.8 kb. So vector is 3.8 kb. And when you introduce the insert of 2 kb, the total size of the recombinant plasmid is 5.8 kb. So yes, this statement is correct. Now please note, this is not the answer because we have to choose the incorrect statement. You have to keep this point in mind. Then statement 2, digestion with BAMH1 will yield one 3.8 kb and one 2.0 kb fragment. So let me use another ink. So if we cut the vector from here, BAMH1, and so BAMH1, two BAMH1 sites are there. So if we cut it from here, we should get one fragment from here to here. So this is around 2 kb. And this is the vector. So from here to here, this is the vector. So yes, it is 3.8 kb. So this statement is also correct and cannot be the answer. Now statement 3 and 4, just have a look carefully. Digestion with eco R1 and Hind 3 will yield 1.7 kb and one 4.1 kb fragment. And digestion with eco R1 and Hind 3 will yield one 2.1 kb and one 3.7 kb fragment. So let's find out. So let's use another ink color. So if we digest with eco R1, that is now if we perform the digestion over here and with Hind 3, so it will cut over here. So we'll get one fragment which is around like 1.2 plus 0.9, that is around 2.1 kb, and one 4.1 kb fragment. So that means this 0.8 plus 2.9, and so 0.8 plus 2.9 gives you 3.7 kb fragment. So basically we are getting the fragments as given in option 4, right? So if the insert is added in opposite orientation on performing the digestion with eco R1 and Hind 3, so these are contradictory statement option 3 and 4. So we will be getting 2.1 kb and 3.7 and not 1.7 and 4.1. So statement 4 is correct and statement 3 is incorrect, and because in the question incorrect statement is asked, answer is option 3. So practice drawing the figures and labeling the figures, and carefully check where you have to perform the digestion and what size fragments you will get. So we have seen that from these three topics, restriction, digestion, modifying and manipulating enzymes and restriction enzymes, we can expect statement based as well as figure based questions, experimental based questions. So now as you are familiar with the type of questions, prepare the topic accordingly and practice as many questions as possible. So keep practicing, keep learning. Thank you.